Welcome to Chapter 4, Cellular Metabolism. In this chapter, we're going to talk about a lot of different processes that your body undergoes on an everyday basis. So we'll start with metabolism. Metabolism is the sum of all chemical reactions that occur in the body, and then cellular metabolism is actually the sum of all chemical reactions that occur on the cellular level. So there's two basic types of metabolic reactions. Those include anabolism and catabolism. Anabolism, like anabolic steroids, those are things that those are reactions that smaller molecules come together to make a larger molecule. Anabolism, small molecules making a large one, that requires energy. Catabolism is like a catastrophe. Things are broken down. So larger molecules are broken into smaller ones and an energy release occurs here. So in anabolism, it requires ATP is made during catabolism, and it provides materials for maintenance, cellular growth, and repair. So anabolism builds up the body. Catabolism breaks down larger molecules, like food molecules that you eat, breaks it down into smaller molecules that you can then use to make your body. So an example here is dehydration synthesis. Now the word dehydration, as we know, means to take water away. So in dehydration synthesis, you put two molecules together. This is two monosaccharides, which simply means a simple sugar is added to another simple sugar, and water is taken away. So here's the water molecule, and so in the end, you end up with a disaccharide, which is just a more complex sugar, and you have a product of water. So you make water there. So this is more anabolism, and that is putting things together to make larger molecules. This is catabolism. So in catabolism, you use the process of hydrolysis. Hydrolysis is using water to break down other molecules. So it's used to decompose carbohydrates, proteins, and lipids. It splits substances, and it's the reverse of dehydration synthesis. So here you've got dehydration synthesis where you make water, and then you go in the opposite direction, and that is catabolism. So you've got a larger sugar molecule here, you add water to it, and you break it down. Okay, this is showing the same thing. Catabolism is taking a large molecule and going in the opposite direction and breaking it down into smaller molecules. Here you see a dipeptide molecule. We're going to talk about this a few times throughout the lecture. A dipeptide is simply two amino acids that are joined with a peptide bond. Peptide bonds join amino acids together, and so you take that the dipeptide and you break it into the two individual amino acids. And in order to do that, it takes water to split the molecule. Now, control of metabolic reactions. We are constantly controlling these things, catabolism and anabolism, in order to maintain homeostasis in the body. We want to keep all processes within normal limits in the body to prevent disease conditions. And in order to do that, we have something called enzymes. Enzymes control the rates of both catabolic and anabolic reactions. Now, what are these enzymes we speak of? Enzymes are extremely important in our bodies. Enzymes are actually proteins, and they catalyze specific reactions. So that means that they are present in order to lower the activation energy that's necessary to start the reaction. So enzymes allow the reaction to actually occur. If we didn't have enzymes, we could sit around forever and the reaction wouldn't actually occur because it takes too much energy to start. Sometimes we may feel that way when we are ready to start studying. We need an enzyme to lower the amount of energy that it's going to take us to get busy, right? So enzymes are actually a very key point right here. They are not consumed in the reaction, so you can use them over and over again. And enzymes are very specific to a particular substrate. They have the ability to recognize that substrate. Now, look right in this area. You have the substrate, that is the thing upon which the enzyme will act. And then you have the active site in the enzyme. So the substrate comes together in the active site to make the enzyme-substrate complex. It is denoted ES, enzyme-substrate complex here. And then you produce a product. And at the end, you will see that the enzyme that was used in the reaction remains unharmed, so it can be used over and over again. The shape of the enzyme is very, very important because we said it's very specific. But the shape of that enzyme can be altered due to excess heat. So if you heat up a reaction, you can break down the enzyme. So it's just like using excess heat to break down any protein. For example, insulin. Insulin exposed to excess heat will no longer be insulin. It can no longer work in the body as such. Radiation, electricity, chemicals, extreme pH values, so being in an extremely acidic environment. Some poisons can also alter that enzyme. Now, denaturation. If something is denatured, it means that it's no longer able to work. A metabolome includes all molecules that are part of metabolism in the cell, and then cells come together to make up tissues, tissues come together to make up organs, and organs come together to make up the organism. So metabolomes are all small molecules involved at any level in the metabolic process. Another name for them are metabolites. Human cells have at least 2,500 metabolites, but less than half have actually been identified. Now, metabolic pathways. As we said, we have enzymes that control metabolism throughout the body. And in order to name an enzyme, it always ends in ACE. Anything ending in ASE is an enzyme. And usually it's got the name of the substrate as the root word. Let's say lactase. Lactase is the enzyme that breaks down the milk sugar lactose. And you know, if you are lactose intolerant, that means that you cannot drink milk sugar because it will just sit in your body. As I said, it will not be able to undergo the breakdown. It will sit in your tummy and it will actually ferment, and the result will be extreme gas, bloating, and diarrhea. So lactase breaks down lactose. Here's another one that's kind of strange. It doesn't involve the root word. This example is catalase. Catalase is an enzyme that breaks down hydrogen peroxide. And we know that hydrogen peroxide is broken down to produce oxygen. That's why you use hydrogen peroxide on different things like your teeth. So whitening strips include hydrogen peroxide, and you see the bubbling that occurs, and that is the enzyme catalase breaking down the hydrogen peroxide to release oxygen to cause a whitening process. More on metabolic pathways. We have all different sorts of pathways, some that are called positive feedback and some that are called negative feedback, and we'll talk more about this throughout the course. In some pathways, negative feedback in particular, it has a rate limiting enzyme. That means that it sends a signal to your body and it says stop, does not produce any more of that substance. So negative feedback. And we'll study that in relation to your thyroid gland. When your thyroid gland has made enough hormone, it receives negative feedback, and enzymes and hormones say, I got enough, stop, and it brings you back to normal. And that's the way that metabolic pathway is controlled. Cofactors and coenzymes. A cofactor is a necessary part of enzymes, and some examples include copper, iron, and zinc. And many organic molecules act as cofactors, and most vitamins are also coenzymes, and they're also called cofactors. So vitamins can be called coenzymes or cofactors, and we need these vitamins because they act as cofactors or coenzymes in order to allow metabolic reactions to take place in our bodies. And if you're missing a vitamin, then that metabolism will not take place. Energy. The definition of energy is simply the capacity of matter to change, the ability to do work. And common forms of energy include many different things: heat, light, sound, mechanical and chemical energy. And we remember from elementary school, energy cannot be created nor destroyed, but it can be changed from one form to another. So we're going to talk about cellular respiration and the use and production of chemical energy in the body. So first we're going to mention the molecule adenosine triphosphate. We always refer to it as ATP. It carries energy in a form that the cell can actually use. So it's made up of adenosine, ribose, and then 3 phosphates. And in order to give you energy, this energy bond here between the second and third phosphate is broken right in this area. When you break off this third phosphate, it provides energy for the cell because that is a very high energy bond right there. So we go from ATP, break off this third phosphate, and we end up with ADP. The breaking of that phosphate itself provides energy. So ATP, when it loses that last phosphate, it becomes ADP, as I just said, and that is called adenosine diphosphate. And it goes back and forth between ATP to ADP. Whenever you add a phosphate to it, you get ATP. When you take off this phosphate, you get ADP. So adenosine triphosphate, break a phosphate, you end up with adenosine diphosphate. 3 down to 2. Now, this is my little sad joke. Where is energy stored? ATP. TP, energy. See, lots of great energy in there. I would love to stay in that TP. It's beautiful. Look at that. ATP. Energy is stored in ATP. Now, release of chemical energy. Chemical energy is released in cellular respiration from molecules, and it's transferred into other molecules. So energy is held in chemical bonds. So what is going on in cellular respiration? We are breaking some bonds and forming other bonds. Cellular respiration occurs in 3 different phases. One is glycolysis, next is the citric acid cycle, and next is electron transport chain. Glycolysis is anaerobic. Anaerobic means it does not require oxygen. It takes place in the cytoplasm, and what is actually occurring is you take one glucose molecule, which is a 6-carbon molecule, and you just break it in half into 2 pyruvic acid molecules. I hope that you are remembering this from the detailed lessons that you had on this in biology class. The next step of cellular respiration is the citric acid cycle. In this class, we simply want to know what goes in, what comes out, and where it occurs. Okay? Citric acid cycle. It is aerobic. It requires oxygen, and carbon dioxide is released. The electron transport chain is also aerobic. Oxygen is required here. And that's the last step of cellular respiration. Arsenic is a chemical that we know as a poison, and it actually works by preventing the cell from extracting energy from glucose. Sorry, that should be from glucose. And so it stops cellular respiration. When you can't extract energy, your cells die because there is no energy available. Okay, so parts of cellular respiration. Okay, cellular respiration of glucose requires glucose and oxygen. So what did we say to start with? It is the process of taking a 6-carbon glucose molecule, breaking it in half to begin with, and then you're producing energy and oxygen, using oxygen to continually produce that energy. So products of cellular respiration: carbon dioxide, water, and energy, which is stored in ATP, and you produce heat. So we said it requires 2 types of reactions, aerobic and anaerobic. Aerobic requires oxygen and makes the most ATP, and it occurs in the mitochondria. Anaerobic does not require oxygen, but it still actually makes a little bit of ATP, a little bit of energy, and it occurs in the cytoplasm. Anaerobic occurs in the cytoplasm. Aerobic, again, when you do aerobics, it takes oxygen. And then anaerobic means the